Okay, we're going to go ahead and get started. Thank you, everyone, for registering and being on this webinar today. I'm Grace Saba. I'm an assistant professor at Rutgers University and Ocean-Acidification Innovation Lead for the Mid-Atlantic Regional Association Coastal Ocean Observing System, or MARACOOS. I also co-coordinate the Mid-Atlantic Coastal Acidification Network, or MACAN. MACAN's other co-coordinator, Katie Goldsmith, who's the project manager at the Mid-Atlantic Regional Council on the Ocean, or MARCO. She's in attendance today, and together we can take any MACAN-related questions at the end of today's webinar. I am very happy to introduce, oops, hang on, it's not changing slides, 1 second. There we go, sorry. I'm happy to introduce our presenter today, Marjorie Friedrichs. She is an associate professor of marine science at the Virginia Institute of Marine Science at the College of William & Mary. She studies the effects of climate change and other human impacts on coastal and estuarine systems through the use of coupled biological physical models. She is currently working on a wide variety of projects in our Mid-Atlantic coastal ocean funded by EPA, NASA, NOAA, and NSF. Today she'll be presenting one of the projects titled Multiple Stressors in the Chesapeake Bay, What Drives Changes in the CO2 System. This is a collaborative project being led by Elizabeth Shadwick at VIMS and also involving co-PI Ray Najjar at Penn State University. So during the presentation, all attendees will be muted. However, we encourage you to ask questions using the question box in your control panel. These questions will be addressed during the 15-minute Q&A period at the end of the presentation. So a reminder that this webinar is being recorded and it'll be available on the MACAN website on the resources page with our previous webinar series. So this is our second webinar in our second series. So coming up in February, we have one that I will be presenting on Research Priorities Working Group. We have two working groups through MACAN. One of them is the Research Priorities, and the second one is the Monitoring Plan Working Group, which is led by Katie Goldsmith, and that'll be in March. So we do have two more coming up in this webinar series. So without further ado, I'll hand it over to Margie. Thank you so much, Margie, for presenting today.
You bet. Thank you for that introduction. It'll work. Okay, so I think you should see the slides. Hopefully you all can hear me okay. So thank you, Grace and Katie, for inviting me, and thanks to everyone else who's tuned in today. I'm relatively new to MACAN, but I'm interested in learning more about what others are doing in the Mid-Atlantic region, and I'm hoping to become more involved in the future. As Grace just mentioned, my background is in coupled physical biogeochemical models, and until recently my work was really focused more on the organic side of biogeochemistry and specifically hypoxia. But a few years ago, Elizabeth Shadwick, who had recently joined our faculty at VIMS, approached me to see if I wanted to team up on a proposal to study inorganic carbon cycling in the Chesapeake, and I jumped at the opportunity. So today I want to tell you a little bit about this, our work together on this project called Multiple Stressors in the Chesapeake Bay, What Drives Changes in the CO2 System. And I also want to acknowledge the work of Pierre St. Laurent and my student Faye Da at VIMS. And also, as Grace mentioned, Ray Najjar and Maria Herman are also from Penn State are also involved in the project. And although this work is funded primarily by NSF, this is also very closely related to some of our other NOAA projects as well. So I want to acknowledge that funding too. And finally, my email address is down here at the bottom of this first slide. So if anyone has any follow-up questions, really please don't hesitate to contact me at any time. So as many or all of you are probably aware, the Chesapeake Bay Watershed, which encompasses parts of 6 states and Washington, D.C. as well, has really seen large changes over the past century, including a population increase from 4 million people in the early 1900s to over 17 million now. And runoff from this large watershed feeds into the Chesapeake Bay, which is one of the largest estuaries in North America. And over this past century, the region has also seen significant increases in nitrogen fluxes to the watershed, largely a result of this urbanization, including pollution from wastewater treatment plants and stormwater runoff. And in addition, there's been an increase in nutrient inputs from agriculture. For example, here on the right, in the figure on the right here, I have a figure showing the increase in the production of manure, the triangles here, together with the increasing atmospheric deposition of nitrate and ammonium, shown by the dotted and dashed lines. But really the biggest change shown here on the figure is the increase in fertilizer on cropland, which is the dashed dot line here. And it really took off in this region after World War II. So these increases in nutrient fluxes, both due to urbanization and increased fertilizers, really have had dramatic impacts on Chesapeake Bay. So just as a reminder and as a background here, when nutrient inputs to the bay are high, for example, from increases in fertilizer inputs, as I just showed in the previous figure, this really stimulates phytoplankton growth in the bay, which eventually sinks down to depth, where it's remineralized and respired, causing oxygen levels to decrease and resulting in increased hypoxia, which is really very damaging to living resources in the bay. At the same time, this process also increases CO2 concentrations at depth, reducing pH and increasing acidification. So although management efforts in the bay historically have really focused primarily on how increased nutrient inputs result in increased hypoxia in the bay, recently there's been more emphasis on how increased nutrient inputs are also causing increased acidification in the bay. So that's really what the talk today is going to be focused on. So just as a reminder, also, at the same time, over the past century, atmospheric CO2 concentrations have, of course, dramatically been increasing as well, also causing increases in acidification in the bay. So there are really these two compounding effects in this region and other coastal regions as well, both leading to increased acidification. There's this local anthropogenic change caused with these increased terrestrial inputs, as well as the global anthropogenic changes, basically the increased atmospheric CO2. And it's really important to tease these two apart, since local and regional managers may have the ability to manage and change local nutrient inputs, whereas they have much less ability to impact global change, such as increased atmospheric CO2. So managers would really like to know how much of the acidification occurring in the bay is due to local anthropogenic effects. So with that in mind, the overarching goal of our project is really to try to distinguish impacts on CO2 variability in the bay due to local anthropogenic changes, essentially nutrient inputs from the land, and changes, essentially increases in CO2 and temperature in the atmosphere. So to address this goal, we're using CO2 system observations to calibrate numerical models, and then conducting numerical modeling experiments to tease apart and isolate the influences of local versus global change. So quick outline, and my talk today is going to be divided into three parts. I'm first going to talk about the CO2 system data that we're using in the bay. Essentially, we're using the long-term 30-plus year EPA time series of water quality data in the Chesapeake, in conjunction with some high quality, or what we call climate quality data, that Elizabeth Chadwick has been collecting for the past couple of years. This includes additional cruise measurements, as well as high frequency underway observations and high frequency buoy observations. So then I'm going to transition into the modeling component of the talk and discuss results of our 35-year control run. And in particular, I'm going to be focusing on the DIC, or dissolved inorganic carbon budget. Essentially, we want to know how much of the inorganic carbon coming into the bay from the rivers is being exported to the shelf, how much is being respired locally, and then how much is being outgassed to the atmosphere. So then I'm going to talk a little bit about our comparison of the early years of the 1900s with the early years of the 2000s, so a century apart here, and shows some results of our experiments aimed at isolating the effects of local versus global change. And finally, I want to end by just talking a little bit about some of our ongoing short-term forecasting efforts. For now, these are involving primarily hypoxia and oxygen, temperature, and salinity. But soon, hopefully, we're going to be putting online some of our short-term forecasts of acidification metrics, such as the aragonite saturation state and pH, and hopefully making these available to the public as well. So that's a little kind of separate, but also I just wanted to mention that to this group since I thought there might be some interest in that as well. So first, the CO2 system data component of the talk. Now, this is really Elizabeth Chadwick's portion of the project, so I'll try to do it justice and answer questions as best I can if there are any. So To start off, as part of the Chesapeake Bay program's water quality monitoring program, cruises have been conducted throughout the bay for the past 30 years, so basically since 1985, with cruises twice a month in the warmer months and once a month in the colder months. So this ends up being about 17 or 18 cruises per year. And a wide variety of water quality measurements are taken. This includes temperature, salinity, oxygen, various inorganic and organic nutrients, chlorophyll, as well as pH. And these cruises sample about 35 main stem stations, showed on the right here, as well as 60 or more tributary stations, although here in this project, we're really focusing a bit more on the main stem. So this is really an amazing data set that we have for the Chesapeake Bay, and these data are really the reason we've been able to develop and evaluate our coupled hydrodynamic biogeochemical model of the bay. But to supplement these data, for the past couple of years, Elizabeth's been participating on these Bay program cruises roughly every other month, so about 6 cruises per year. She's been going out in her lab group and collecting both TCO2 and alkalinity. So she collects the samples on board, and then her group analyzes them later at VIM. So in this way, she's able to produce climate quality data sets of not only TCO2 and alkalinity, but she also gets pH, PCO2, and carbonate saturation state. So part of the goal of this data collection has really been to investigate the quality of the simultaneously collected Bay program water quality pH data. Since before we use this 30+ year time series of pH to look at long-term trends, we really need to look at the uncertainty in the water quality pH data. So how do these two types of pH data compare? Well, here we have a very preliminary analysis of this comparison. On the y-axis, we have the Bay program's water quality pH data, and then on the x-axis, we have Elizabeth's higher quality or so-called climate quality pH estimates computed from her TCO2 and alkalinity data. So as you can see, the comparison for this past year is really quite good, though we do see that the Bay program's pH data tend to underestimate pH a bit, the difference between the two being greatest at lower pH values, generally in low salinity waters and in the summer. But in general, except for a few outliers, really the comparison is looking very promising. So we're excited about that. So in addition to the new climate quality cruise data, Elizabeth also started underway PCO2 sampling in the Virginia portion of the Bay. So basically, she collects high resolution, basically every 2 minutes, temperature, salinity, and PCO2 data. And this is particularly helpful in that it allows us to interpolate between the discrete stations data that we have for the CO2 system parameters that I mentioned earlier. So here on the left, I have an example of a figure of showing underway PCO2 data from May 2017. And you can see the spatial variability with lower values, so to the south of the Potomac, near the Rappahannock. And then you can also see in regions where the cruise track repeats itself, how quickly POC concentrations can change, go from green 400 up to 550, perhaps even changing with a tidal cycle. So these are some of the interesting features of the data that we're analyzing now. So finally, the other type of high frequency data that we've begun to collect in the Bay is from an autonomous CFOX sensor that Elizabeth's put with the help of NOAA and CBNEERS on the York Spit CBIBS buoy. So for those of you not familiar with CBIBS, this is the Chesapeake Bay Interpretive Buoy System, which includes 10 buoys spread throughout the Chesapeake Bay and its tributaries. And each buoy collects a suite of hydrodynamic, water quality, and atmospheric data. Now here on the York Spit buoy, we also have pH information as well. So here I have example plots showing data from the CFOX, temperature, salinity, pH in the upper right, and oxygen. And these are from the first 2 deployments of 2017. So these are hourly data. You can see the high frequency variability in each of these fields, particularly in the pH, constant pH over here on the upper right. Also, you can see the relatively smooth transition between the 2 deployments, which we were very happy to see. The first deployment is shown shaded in gray, and white is the second. So this is all very exciting. A lot of data here that we're really beginning to analyze. And maybe at a future call, Elizabeth can talk a little bit more about some of the interesting results she's finding from these data sets. So essentially, that's it for the first part of my talk on the historical and new CO2 system data that we have in the Bay. And now I'm going to move on to the CO2 system modeling component of the talk. So essentially here we're using the data that I've just presented to you in order to calibrate and evaluate our coupled hydrodynamic biogeochemical model. However, I will say that since a lot of the Elizabeth's new data are really just coming online and have only been available for the past year or two, most of the model development and calibration that we've done over the past many years has really been done with the Chesapeake Bay program 30+ year water quality data set. So I'm going to first talk to you about our control run, which is from 1980 to 2014, and how the inorganic carbon budget looks for those years, and then discuss again our experiments where we're teasing apart the impacts of local and global change. So first, a little bit about the model. So the model we're using for the project is a fully mechanistic linked land estuarine ocean modeling system, and it includes both carbon and nitrogen cycles. The terrestrial model is called DLEM, which stands for the Dynamic Land Ecosystem Model, and it's run by Han-Chin Tien and his colleagues at Auburn University. DLEM is run from the late 1800s to the present and is forced by data on land cover from the National Land Cover Database for the recent years, and then for the historical years, they actually use county-level data. The climate forcing, basically temperature and precipitation, comes from the PRISM database. And then the output from this DLEM model is then basically used as the input to the estuarine model. So this provides the riverine inputs, which are so critical to the estuary. And so our estuarine model is an implementation of ROMS, which stands for the Regional Ocean Modeling System. And the biogeochemical component of the model has been specifically developed for estuaries and is called ECB, which stands for Estuarine Carbon Biogeochemistry. And we're working currently on nesting this ROMS simulation inside a larger shelf implementation of ROMS developed for the US East Coast called US ECOS, but that work is still really in progress. So right now we're just nudging to climatological values on the continental shelf. So just a few other details about the models. Again, DLEM is this fully mechanistic model, includes both the hydrology and biogeochemistry of the Chesapeake watershed, and provides us with daily organic and inorganic carbon and nitrogen inputs to the estuarine model. And in the estuarine model, ChesROMS-ECB has a spatial resolution of about 1 kilometer, has 20 depth levels. It also includes tides, which of course are critical in this region. And like DLEM, it includes a full suite of biogeochemical state variables, not only phytoplankton and zooplankton, but various forms of nitrogen and carbon, inorganic and organic. And this also includes dissolved DIC, dissolved inorganic carbon, alkalinity, and PCO2. And of course, from these, we can calculate saturation state and pH from the model as well. So in terms of skill assessment for the system, DLEM has been evaluated with discharge and nutrient data from individual USGS gauges throughout the watershed, as well as regression estimates from the USGS LOTUS. We've also done quite a bit of skill assessment on the ChesROMS-ECB estuarine model using the in situ water quality cruise data that I mentioned before from the Chesapeake Bay program. And I don't have time to go into all the skill assessment details today, but the details have been published in a few of these citations listed here at the bottom. And a number of these actually appeared in a special issue of JGR Biogeosciences devoted to carbon and nitrogen fluxes at the land-ocean interface. So if folks are interested in more information, that would be a good place to look on that. However, since actually much of our published estuarine model data comparisons have really focused more on the nitrogen cycles and the carbon, I thought I'd include this slide here showing a 30-year climatology from 1985 to 2014 of PCO2 model output for March on the left here and September on the right. And the model output is shown as contours in the background, and then the circles, individual circles on each map represent the Bay program's data climatology. And the point here is to simply show that the model does a reasonable job of reproducing climatological PCO2. Both the model and the data show uptake in the spring, here on the left, and mostly outgassing in the fall, here on the right. And both the model and data also show similar spatial variability, with the lowest PCO2 values really appearing in the mid to lower bay, generally away from sources of higher PCO2 water. So as I mentioned before, one of the things we want to do with this modeling system is to get a better understanding of what happens to the inorganic carbon that comes into the bay from the rivers. So essentially, the model computes how much of the inorganic carbon from the rivers is converted to organic carbon through net ecosystem production, by which I mean production minus respiration, or NEP. I'm going to be using that abbreviation here. And then how much escapes to the atmosphere via air-CO2 exchange, and ultimately how much is exported to the continental shelf. So basically the source of DIC here are the rivers, and then there's three places that this inorganic carbon can go, either to NEP, outgassed to the atmosphere, or exported to the shelf. And then when we run the model over multiple decades, we can see whether there is any evidence for changes in the magnitude of these processes. For example, whether there might be any increase or decrease in air-CO2 flux over the past several decades. So on to some modeling results. Here I'm showing our 35-year time series of model output from 1980 to 2014 of the input of river DIC in blue at the top, and then the ocean export of DIC in pink at the bottom. So the signs can get confusing here, but essentially I'm using convention that positive values represent a flux of DIC into the estuaries. So here the rivers are bringing DIC into the system, so the blue line is positive. And negative values represent sinks of DIC. So in this case, the export of DIC is to the coast. So this is negative, a sink of DIC. So clearly, these two time series, the inputs from the rivers and the export at the bay mouth, are opposite in sign, and they actually track each other very well, as you can see, and are very highly correlated. So essentially, when there's more water coming into the bay, there's also more water exiting the bay. However, the DIC concentrations of the water coming into the bay are actually higher than those of the waters exiting the bay. So the magnitude of the river blue line here is greater than that of the pink line below. So what we really are interested in here is the difference between what's coming in and what's going out. So that's what I'm showing here, and I'm going to show for the rest of the talk. I'm going to concentrate on. I'm going to call this the lateral flux, basically the rivers minus the export. So here we see that on average, the net lateral input of inorganic carbon, essentially that coming in from the rivers minus what's going out to the ocean, is roughly about 0.3 teragrams carbon per year, though this can vary actually between about 0 up to about 0.8. So what happens to this inorganic carbon entering the bay? Again, there are these two possibilities. It can be turned into organic material through net ecosystem production, or it can be outgassed to the atmosphere. And there's actually a third possibility. It could be that the inorganic carbon concentrations in the bay are simply increasing in time, but this is not happening, and we aren't going to consider that any further here. So on this slide, I've added a red line denoting NEP. So plotted here is a negative value since it represents a sink of inorganic carbon, so it's actually negative NEP. So here we see that production is always greater than respiration in the bay. Basically, NEP accounts for a large amount of the incoming DIC, but not all of it, since you can see the red line is less than the blue line. And in particular, it's much less variable. From year to year, NEP is relatively consistent, whereas the lateral inputs of DIC really vary dramatically from one year to the next. So what are we missing here between since the red line doesn't equal the blue? Well, again, the other term here is the air-sea flux of CO2. And here on this figure, I've added it as a black line. And you can see that although in general, the air-sea flux represents a sink of DIC, essentially the bay, so it represents a sink. It's generally below the x-axis. Basically, the bay is outgassing CO2 to the atmosphere, and that's denoted by the black line being generally below the x-axis. At times, it does get above the x-axis and positive value, and that means that there's net uptake. The bay is providing a source of DIC. The air-sea CO2 is a source of DIC for the bay. So if you squint your eyes a bit, you can see that at times when the river inputs of DIC are particularly high, and outgassing is also high. But so for instance, here I can use the pointer, I guess. When the bay, when the river minus export, the lateral inputs are high. Also, the outgassing is high, and when the river inputs are low, the outgassing actually turns to uptake. So what's causing this? So it turns out that whether the bay acts as a source or sink of DIC to the atmosphere depends really on whether a particular year is dry or wet. So for example, during these dry years that I've circled here, we have relatively little precipitation, and the net input of DIC to the bay is very low. The blue line is very low, close to zero, and as a result, the bay ends up taking up atmospheric CO2. In contrast, when we have very wet years with high precipitation, as I'm highlighting here, we have high river inputs of DIC, and in this case, the bay outgasses CO2 to the atmosphere. So overall, we see that natural interannual variability in the bay is primarily governed by precipitation. So in wet years, we have more carbon inputs, which results in more CO2 outgassing, whereas net ecosystem production really changes much less between the wet and dry years. The red line is much more constant. So now I want to get back a little bit to the overarching goal of the project that I mentioned before, which again is to distinguish the impacts of local versus global anthropogenic change. So given the large amount of natural variability between the dry and wet years that I just showed you, this is really quite challenging. So essentially, on top of all this natural variability, human activities are causing significant changes to the system as well. So specifically, human activities have caused increasing DIN loading, as I mentioned earlier in the talk, due to changes in land use and fertilizer and urbanization. And in addition, over the past century, alkalinity and DIC loading have also been increasing the Chesapeake rivers, and we think this is primarily due to acid mine drainage. And then in addition, we have the global changes in terms of increasing atmospheric CO2 and increasing temperatures. So really the question here that we want to use the model to address is, over the past century in the Chesapeake, how has inorganic carbon cycling changed due to human activities? So this is a complex problem, and although we're working on all different facets of this, today I really want to focus specifically on two particular human activities. Basically, I'm going to focus on increased nitrogen loading and increased atmospheric CO2. And these other issues, basically the increasing atmospheric temperature and acid mine drainage, are other things that we're currently working on, but I'm not going to be talking about today. So in our model experiments, we're going to compare 3 model runs, one for the early 2000s, so specifically 2000 to 2014. And this is basically the same as the control run, which I was already describing to you. And then the second model run is exactly identical to the first, except that we use atmospheric CO2 and riverine DIN inputs that are specific for the early 1900s. So other than this, again, the model runs are basically the same. So finally, we have a third model run that we will call no atmospheric CO2 increase, since here we do change the riverine nitrogen concentrations as in the previous second model run. But we don't change the atmospheric CO2. So those stay at the 2000 levels. So this third run basically can help us understand how much of the difference we see between the first and the second run is due to local nitrogen input changes rather than global atmospheric changes. And just to remind you of the differences between these runs, the upper two figures here show how riverine nitrogen and atmospheric CO2 has changed over the past century. So you can see both and then here I have shaded the first 15 years that we're using for the one simulation, the first 15 years of 1900s and the first 15 years of the 2000s. So you can see really the dramatic increase in riverine nitrogen between the 1900s and the 2000s, and also the increase in atmospheric CO2 between the early 1900s and the early 2000s. And again, these are the two differences between the model runs. Everything else we're holding constant. So I'm now going to show you the average seasonal fluxes, so basically the climatological fluxes for these 15 years for these model runs. So starting with the results for the 2000s, here in this first figure, you can see the lateral fluxes, once again in blue, the NEP in red, and the ARC flux in black. I'm trying to stick with the same color scheme here to make it a little easier. Here we see in this contemporary time period, we have a large amount of production in the spring, causing spring uptake of ARC CO2. So we've got this large production accompanied by this uptake of CO2. And this occurs in March. So then later in the summer, we can see these two switch directions. So we've got respiration exceeding production. So NEP is above the x-axis here. And at the same time, we've got outgassing. So the black air-sea flux line dips down below the x-axis. So with these red and black lines being roughly inversely correlated, we can see that the seasonal variability is really here is really being driven by biogeochemistry rather than the physics. So in this next figure, we have the results from the early 1900s. Now, in comparison to the results I just showed you for the 2000s, and we can flip back and forth between that's for the 2000s, and this is for the 1900s. Again, 2000s, 1900s. You can see that the lateral fluxes are actually very similar, but we see less production in the spring and also less CO2 uptake in the spring as well. So right here is where we see the biggest differences. In the fall, there is some differences, but these differences are a bit smaller. So the nice thing about using modeling experiments is that we can answer the question as to why, at least in the model, we're seeing these differences. So basically, the question is the reduced CO2 uptake in the spring due to the higher atmospheric CO2 concentrations, or is it due to the higher NEP resulting from the higher nitrogen inputs? So to address this question, we look at our third model run where we don't change atmospheric CO2, but rather we just change the nitrogen inputs. And those are the results shown here. So again, if we flip back and forth, that has both atmospheric and the nitrogen inputs, and this just has the nitrogen inputs. You can see that the changes are really very small between these two model cases. So this demonstrates that most of the change in the outgassing that we're seeing between the 1900s and the 2000s is actually due to changes in the nitrogen inputs, riverine inputs to the model, and not due to the change in atmospheric CO2. So to look at this quantitatively, finally, in a little simpler manner, perhaps, this is a bar plot shown again for the early 1900s on the left, the 2000s on the right. And again, we have the same color coding with the lateral fluxes of DIC shown in blue, net production minus respiration shown in red, and air-sea fluxes shown in black. And overall, we can see almost a doubling of NEP between the 1900s and the 2000s in this red bar here, which is essentially entirely caused by the increases in nitrogen inputs to the bay. So that is local anthropogenic change. And then the black bars decrease even more substantially, actually, by pretty much a factor of 4. And it turns out that our experiments are showing that about a third of this difference between the 1900s and the early 1900s and the early 2000s is due to the global changes of atmospheric CO2, and about 2/3 is due to the increased nitrogen inputs. So I just want to briefly summarize these modeling results. They were kind of complex, and I went through them quickly, so hopefully they were clear. But just to summarize this component of the talk, in terms of interannual variability, we saw the natural variability of the system is really controlled by whether a particular year is wet versus dry, so essentially how much precipitation has occurred in that year. And we see that NEP is surprisingly constant regardless of whether a particular year is wet or dry. However, we do see that in the wet years, we have much more outgassing of CO2 to the atmosphere, and in dry years, we have much less outgassing, in fact, potentially even uptake of atmospheric CO2 by the bay. In terms of seasonal variability, we see that these are really, possibly not surprisingly, controlled by the biogeochemical processes. We see uptake of CO2 during the spring and outgassing for the rest of the year. And then in terms of century scale variability, in the 2000s compared to a century earlier, we really are seeing higher NEP, and this is due to increased nitrogen inputs from the rivers over the past century, largely again due to these fertilizer inputs. And we're also seeing less CO2 outgassing to the atmosphere than we did in the early 1900s. And about 2 to 1, this is due to the increased nitrogen inputs in the bay, and only about a third is due to the increased atmospheric CO2 concentrations. So basically, well, hopefully I now have just a few more moments to talk a little bit about our short-term forecasting system. And I know I don't have a lot of time, so I'm going to make this relatively quick. But again, if folks have other questions, I guess there'll be some time today. And also don't hesitate to follow up with me for if you want any additional information. So for the past couple of years, thanks to some NOAA funding from the Coastal Ocean Modeling Testbed Project, we've been using our modeling system to produce short-term forecasts for the Chesapeake Bay. So, so far, we've been concentrating primarily on hypoxia forecasts, but as part of the modeling system, we're also generating forecasts of acidification metrics, including DIC concentrations, alkalinity, PCO2, and pH, and saturation state. We just aren't quite ready to post these online yet. And this is partially because we're still really working to establish stakeholder focus groups in the regions that include basically the folks that are interested in using these types of products. So I'll just say right up front that if anyone on the call has an interest in these forecasts, acidification metric forecasts, or knows of industry folks that might be interested in having access to these forecasts, please go ahead and let me know. So our forecasts of oxygen, temperature, and salinity are online at the website listed here at the top of the page, www.vims.edu/hypoxia. So this works well. This site can work well both on your computer, desktop, laptop, as well as mobile phones, which is really an important consideration for our end users who are primarily recreational and commercial fishermen. So a lot of these folks want to check the hypoxia forecast when they're out on the dock before taking their boat out. So it was important that these be reliable on mobile devices as well. So if you went to this website back on July 28th, this is on the figure on the left is what you would see. You can actually go to it right now as well. It's a very boring plot because the waters are very, very cold here and very, also the plot is just entirely blue. There's no hypoxia problems in winter here in the Chesapeake Bay. But again, so if you went to this website back on July 28th, this is what you would see. This figure represents bottom oxygen for this particular day, July 28th. And here we've color-coded the healthy waters as blue. So this represents basically good bottom water, which is healthy for bottom-dwelling fish and crabs. The red and orange colors denote regions where bottom oxygen is very low, where you might not find any fish or any live crabs at all. And the yellow and green is somewhere in the middle. So on this site, we also post not only the forecast, as I showed you, same on the left here for July 28th, but also here I'm also showing the forecast for what July 30th would look like if you went to the website on July 28th. So here you can see the low oxygen zone, or so-called dead zone, is considerably smaller on the 30th than it was on the 28th. And in particular, the western side of the bay, right here, is showing considerably healthier waters than the eastern side, and considerably healthier waters than they did 2 days before. So this could definitely be useful information for our stakeholders who regularly want to know where the worst and the best waters are in the bay for the fish, and which areas are getting better from one day to the next and which areas are getting worse. So this summer, these forecasts got actually quite a bit of press in a number of different local papers. And also, I also wanted to make sure to mention that we're actually in the final stages of having these forecasts appear on the Maracus Oceans Map website. So if you look there in the next week or so, they should be appearing on the Maracus website as well. And again, I just wanted to mention that this forecast work to the group here today, because we are also forecasting acidification metrics, even though they aren't posted online yet. Basically, again, we're fine-tuning the calibration and evaluation of the products. And we're also working to identify a stakeholder focus group for these products. So again, this is just a very short introduction to the short-term forecast. But if anyone is interested in additional information, I'd like to encourage them to get in touch with me. And with that, I will take any questions.
Margie, thank you very much. If you guys, anybody has any questions, type them into either the question box or the chat box on your control panel. I haven't seen any pop up yet. I have a quick question though, Margie. As far as the nutrient loading in the Chesapeake Bay, what, if any, regulations are there for that? Do you know that? And if there are regulations, are you using those types of potential reductions into your models?
Yeah, that's a very good question. So this is a huge deal in the Chesapeake Bay. It's all about nutrient regulation. So they're called total maximum daily loads, or TMDLs, and they were set up back in, well, the most recent version was set up in 2010, and we're in a midpoint assessment right now in 2017, and then we will see if we got as far as we were, reduced nutrients as far as we were supposed to it by 2025. That's the ultimate goal. So we do take those into account in a lot of our work, and that is actually in our future work. So what I talked about today was from 1900 to 2000, but we also have another talk, one of my talks is talking about the future for the Chesapeake Bay. And so there we're looking at the impacts of additional sea level rise, climate change, and also looking at what impact we will see from these presumed nutrient reductions. We've made some progress in the past several years towards reducing nutrients, but we still have quite a long way to go before we reach the levels of the TMDLs that are required by 2025. So some of our forecasts go out, or projections, I would say, go out to 2050. And then we look at what is the impact of that change in temperature by 2050, what is the impact of changes in sea level rise, and also what is the impact of, presumably, we will have accomplished all these nutrient reductions as well. So that kind of is definitely something we're looking at. I just didn't talk about it today because it's more of the positive, future nutrient reduction side of the story.
Great, thank you, Margie. Oh, here we go. We've got some questions, but all of a sudden the box just shut. Here's one from Katie Goldsmith. What is the anticipated time frame for getting the near-time acidification forecast available to the public? Will this also show bottom acidification, or will it be elsewhere in the water column?
Yeah, great question. So we can definitely do bottom acidification as well as anywhere else in the water column. We could do the minimum pH, the location of the minimum pH in the water column. We could do a variety of different factors. So in terms of the time frame, we're really want to reach out to stakeholders first in order to see what would be most useful for them. Yeah, hopefully this year. I think in 2018, we're also looking for funding, of course. So that would speed up our process. But yeah, so these are but I'm interested in working, and sometimes in order to find funding, you really have to demonstrate that this would be a useful product for people. So and I was very interested yesterday on the CCAN webinar to hear about what they're doing on the West Coast and how people are using these short-term forecasts on the West Coast as well. So I know they are definitely already being used on the West Coast. They don't seem to exist yet, as far as I know, on the East Coast. But yeah, so hopefully not too much longer.
Great, thank you. There was a kind of a follow-up question for that from Beth Turner. She was asking about, you were working on acidification forecasts and identifying stakeholder groups. I know you just talked about the forecast, but maybe can you elaborate on on identifying the stakeholder groups?
Yeah, so that's where I would love assistance from anyone online that can help us with that. What we've reached out so far to, and Elizabeth, this is mostly Elizabeth's, has been reaching out to oyster hatchery operators in the Bay, and they seem to be the ones that are most interested and feel like these products would be of most use to them so far. But I do think that there are other folks that also that may not be worried about acidification yet, but are likely to become concerned in the near future if they're not already. So I think our end user group and stakeholder group will hopefully expand. But again, if other people have ideas for who might be interested in these types of forecasts, I would be very interested to hear.
Great, thanks, Margie. And me and Katie might be able to help you with that as far as the stakeholders we have involved in MACAN. So we can talk at another time about that. There's one more follow-up question regarding your forecast, so I'll go ahead and ask this first before I move on to other questions. This one's from Beth Phelan. It says, based on what you just said, bottom pH and surface pH are usually different. So how are you handling that in your forecasts?
Yeah, so they're definitely different in our model. And so we can, this is where the stakeholder focus group really helps. And this is in terms of the hypoxia, where we have worked very closely with our recreational and commercial anglers and fishermen to see what type of products that would be most useful for them. So in terms of the hypoxia, it's definitely bottom oxygen that they're really concerned about, or the depth at which oxygen becomes less than 3 milligrams per liter, and that's another metric that figure that we're putting out there for them. So for in terms of the acidification metrics, a lot of it depends on what is of most interest to folks. I mean, we could present maps of bottom pH, we can present maps of surface pH, we could present maps of average pH. Not sure that would be of any use. But yeah, so our pH definitely does vary in the water column, and we can definitely produce side-by-side maps of bottom, surface, or depth of a certain level of pH, say.
Right. Yeah, that'd be really useful, I think, for a lot of the stakeholders.
Yeah.
Okay, so some other questions for different kind of topics. Another one from Beth Phelan. Are the EPA water samples from the surface?
They are they are throughout the water column.
I think she's asking, are the EPA water samples from the surface like the underwater samples? I think she's asking if they're similar.
Mm-hmm. You mean so in the terms of the way they are collected? Yes, that's what I gather. They have I think it's something like a YSI on the boat that they calculate or they measure pH throughout the water column. So it's just the same instrumentation at the surface. Obviously, the values are going to change considerably with depth, but yeah, do you think that answers the question?
I think so. I think I'm not quite sure if she was asking about collection or the actual values, but I think you answered both of those. So
Okay.
Beth, if you're still on and you have a clarification question on that, just type it back in. Another question from Jason Greer. The field data didn't show super strong correlation between DO and pH. Any thoughts about why, given the strong effect of DIN on carb chemistry represented in your model results?
Yeah, that's a very good question. So I'm trying to think of which figure that I showed that DO and pH were not well correlated. Yeah, I think I would assume it would be a time scale issue that if you look at, okay, so this is probably the DO and pH here, where that they're not particularly correlated on that. I think what's going on, well, you do have, of course, the pH is on a logarithmic scale, oxygen is not, and what you're seeing in the oxygen is 90% solubility, or largely solubility, the effect of solubility. So you've got that's going to play a large role, and yeah. So I'm not sure. It's a very good question, and something we're going to have to look into more. We just got these data, so
well,
I would have expected them.
Can you clarify the CBIB CFOX sensor? That's a surface sensor, correct?
It is at the surface, yes.
Okay. Yes. So maybe maybe the correlations would be different at depth.
Yeah, at depth. Yeah. I think you're probably right. Yep.
Okay. Thank you. And there's a few more. Let's see. Vincent Guida, the summer DO patterns in the bay show depression in the mid bay, but not the upper end where Susquehanna enters. How can you explain this?
Okay, so the low DO, the high hypoxia in the central bay. So it turns out, yeah, that's a very good question too. it turns out in the upper bay where the Susquehanna comes in, it also brings in a lot of sediment. So it turns out that the light so here I think like right here in the upper bay, you've got all these nutrients coming in. You would think that you would have lots of growth and lots of hypoxia, but in fact, the Susquehanna brings in so much sediment, it blocks the light and there is not much growth up here. So the growth occurs after the sediment falls out, and then you've got more light up here, so you've got more production, more sinking of the material, and you get more hypoxia downstream and not up in the high up in the tributaries where light is limiting.
Great, thank you. Another question from Carrie St. Laurent. Can the forecast model also assess time length? In other words, how long a certain pH or hypoxia threshold was exceeded?
Yes, in that, you know, our forecast goes out for 3 days. So we can, you know, up to 3, we can't go farther ahead than 3 days. That limit is basically due to our wind, how the NAM Weather Service winds forecast that we use. But we can tell how long certain, at any particular time, how long these low DO or low pH conditions had been occurring at a certain site. Yeah, in the past, up to, you know, so basically we've run this simulation straight through from 2014 through now, so we can tell you how long, you know, what intervals of time these thresholds have been exceeded.
Thank you. Another question from Emily Jo Front. Do you take discrete samples to calibrate the CFOX? How does the sensor behave throughout deployment? Is there significant impact on biofouling? I'm assuming this is a question directed more towards Elizabeth Shadwick, who's unfortunately not able to be here today.
Yeah. So I will say that I think they are taking samples there when they deploy. Again, these 2 deployments were quite long, as I showed there in the figure. I think it went from November to May, and it was 2 deployments, so almost 3 months, and then they took it out and they put it in for another 3 months. So if I recall correctly, November, December, you know, January, then they took it out in February to check on the biofouling. There was very little, if any, so they could have left it in much longer. And then they put a new one out in March. So we're still working on the summer data, but so far in the biofouling in the spring and the winter was not really an issue. And I think, yes, that they are getting, when they go out to service the buoy, they are getting in situ samples. And also we've got the cruises that are not right at the buoy, but not too far away as well, that can help with the in situ sampling as well.
Great, thank you. And I just have a correction. Her name was Emily Jo Frost, not Front. Sorry about that. I read that wrong. Beth Phelan has a follow-up question. She said she was asking about, are the EPA samples through the water column versus the underway samples, which I assume are on the surface? I think it's more the two different methods of collecting the sample. She's asking if they give different results. The underway versus the discrete samples, I think.
Yeah. So then in terms of, so Elizabeth is collecting those high quality TCO2 and alkalinity, and of course she can get PCO2 from those data, and then she has the underway PCO2 data as well. So I think she's making some of those comparisons now. I don't think she's, I haven't seen them yet in terms of how well those compare. And then, yeah, so then the Bay program, the old pH water quality data, that's all the way to the bottom. And again, the pH doesn't match the match is okay with the climate quality pH and the Bay program water quality pH, but we haven't looked at an underway PCO2 compared to the water quality PCO2. So yeah, I would say that these are all pretty new data. It's taken us quite a while to get the recent data from the Bay program from this past summer. So we really have only had the matchups only relatively recently. So I'd say more to come on that.
Okay, great, thanks. And then I just have one more question for you. The CBIBS buoy, there is another one going in to the Chesapeake Bay near the mouth of the bay in a few months, right? So
right.
Be using that for your model input and forecast?
Yes, we absolutely hope to, and yeah. So I think that are you connected with that a little bit?
Only because some of our Marcus partners are involved in that. I'm not directly involved, but just through Marcus.
Yeah.
Right.
So I've heard
it's a new OA buoy with I think it has PCO2 and a CFOX.
Yeah, that will be great. I think for the modeling, what we really need most of all is bottom sensors. So on the York Spit, it's at the surface, as we were mentioning before. And so I don't know whether the OA buoy is going to have something below the surface as well as at the surface. But that's kind of what we're working towards. We're trying to work towards is outfitting. I think it was Weijun Sai had a CFOX at the bottom of the Goose's Reef buoy, which was really helpful for our modeling while that was functional.
Great. Okay, I'll look into that. I right now I'm thinking it's just surface, but I could be mistaken. So I can look into that for you.
Yeah, but and even surface, we'll take everything we can get. So surface is great too.
Great. All right. Thank you so much, Margie. I really appreciate you presenting today, and for everyone that attended, thank you so much. We have, just a reminder, we have another webinar next month, so we'll send out the registration link to that in a few weeks. Thank you, Margie.
Great. Yes. Well, thank you so much.
Okay. Bye, everyone.
Okay. Bye-bye.